Hi everyone. This week in biochemistry, we are going to discuss the electron transport chain. And it is just that. It's the transportation of electrons through a chain of intermembrane proteins. So far in glucose oxidation, we have covered glycolysis, the pyruvate dehydrogenase conversion to acetyl-CoA. We've also talked about pyruvate's conversion to lactic acid. And thirdly, we've discussed the citric acid cycle. So there's one more step in the process of glucose oxidation, and it is that of the electron transport chain. The main learning goal from this module is to be able to describe the transfer of hydrogen ions and electrons in electron transport and the process of oxidative phosphorylation in ATP synthesis. Remember the applicability of this information. Go back to the process of eating food. So we eat food with carbohydrate in it. Then, through the process of digestion, we catabolize the food to its smallest carbohydrate monomer, mostly glucose. Glucose is then in the blood and can be taken up by all the cells within your body through insulin-mediated glucose uptake or contraction-mediated glucose uptake. Now, depending on the energy status within the cell, glucose is either then stored as glycogen, which only happens in the skeletal muscle or in the liver, but most glucose undergoes glucose oxidation to create ATP to undergo hydrolysis to yield chemical energy needed for cell functioning. So it all comes back. Make this information applicable to you. This is that fourth step in glucose oxidation to yield the energy that we need to then break down and create for our own muscle and cell functioning. There are a few things on this slide you need to draw attention to. Firstly is the location of the processes. Glycolysis happens in the cytoplasm or the cytosol. The citric acid cycle occurs in the mitochondrial matrix, and the electron transport chain, as we will learn today, happens in the inner mitochondrial membrane. The second thing is the processes that we have discussed so far, glycolysis and the citric acid cycle, produce smaller amounts of ATP, and this is done through substrate level phosphorylation. Today we will explore the electron transport chain, where there is a high production capacity of ATP from oxidative phosphorylation. Here we will see how oxygen is essential for this to happen. And lastly from this slide, I want you to be able to see how NADH from glycolysis and NADH and FADH2, our coenzyme reducing equivalents, move into the mitochondria to help carry the electrons needed for the electron transport chain. I want you to recall that the anatomy of the mitochondria provides insight on the importance of the function of the mitochondria. There are two membranes that are impermeable to mostly everything in order to protect the energy production that is happening within. This organelle is known as the powerhouse organelle of the cell, and as such, the anatomy dictates how important the functioning is. The electron transport chain occurs along the inner mitochondrial membrane, which borders the inner membrane space between the outer mitochondrial membrane and the inner mitochondrial membrane, and the mitochondrial matrix, which is where the citric acid cycle occurs. Also note that there are many citric acid cycles in the matrix operating at once, and many electron transport chains in the inner mitochondrial membrane operating at once, to ensure we are producing enough ATP to meet the energy needs of every cell. This slide provides an overview of the electron transport chain, and at first glance it may seem confusing, but as each slide goes on, I'm hoping that we are able to delineate each of the steps and help you understand it a little bit better. So the reduced coenzymes NADH and FADH2, which are produced in glycolysis, the pyruvate dehydrogenase reaction, and the citric acid cycle are oxidized. That means they're going to be losing their hydrogen protons and their electrons. And these are lost to provide the energy for the synthesis of ATP. So in the electron transport section of the electron transport chain, also known as the respiratory chain, what we have are hydrogen ions and electrons from NADH and FADH2 are passed from one electron protein carrier or acceptor to the next until they combine with the last electron acceptor, which is oxygen, to form water. What we see is a coupling of the electron transfer with hydrogen proton movement across the inner mitochondrial membrane from the matrix. And this creates an electrochemical proton gradient, where we have a high concentration of hydrogen protons in the inner mitochondrial space and a low concentration of hydrogen protons in the matrix. And so we move from high concentration to low concentration. And so this movement happens through the ATP synthase, and it drives the movement of the protons and allowing for the synthesis of the ATP. This may all seem very confusing right now, but as we proceed in the slides, I'm hoping that it will become more clear. I want you to take a minute right now and watch this video on YouTube on the electron transport chain. I find that it provides a good visual overview of the electron transport chain and the movement of electrons through the chain and protons across the membrane from the oxidation of NADH and FADH2. I'm a visual learner, so this really helps me get a grasp of what actually is happening to understand all the theory that I'm spilling to you in the slides. This is a static diagram of the electron transport chain, and there are a few things I want to draw your attention to. Firstly, the electron transport chain is composed of two major steps. The first is electron transport, and the second is ATP synthesis. Second of all, there are 5 transport proteins embedded in the inner mitochondrial membrane. In the picture, they are indicated by blue, purple, yellow, green, and pink intermembrane proteins. And as we'll see, they are the proteins that transport the electrons and generate energy to pump hydrogen protons across the membrane. The large orange molecule on the far right is the ATP synthase enzyme. It's very big and it's really cool. And it catalyzes the synthesis of ATP by using the energy from the hydrogen proton pump. Lastly, I want you to see the difference in the number of hydrogen protons between the matrix and the inner membrane space, which has the higher concentration. Remember, protons move from a high concentration to a lower concentration, in this case from the inner membrane space to the matrix. They use the ATP synthase molecule as a channel. Before we continue on, I want you to tally the total of net ATP and reduced coenzymes through glucose oxidation so far. Try this on your own first. So what we have is glycolysis. We have a net of 2 ATP and 2 NADH. Oxidation of 2 pyruvate, which is the pyruvate dehydrogenase reaction, we yield 2 NADH. The citric acid cycle with 2 acetyl-CoA, which are produced from 1 glucose molecule, we yield 2 ATP. Well, they're really in the form of GTP, which are equivalent to ATP, and 6 NADH and 2 FADH2. So the total products from 1 glucose molecule so far are 4 ATP, 10 NADH, and 2 FADH2. The reducing equivalents produced for every 1 glucose molecule move to the electron transport chain and are oxidized. That means they lose or donate their electrons to fuel the proton pump, also known as the proton motive force. We'll talk about this more on the coming slides and in the next mini online lecture. Ultimately, NADH and FADH2 are full of energy potential in the form of electrons.